So we had finished watching a video about the membrane potential and how it was created. There are some questions coming up still, maybe one or two, but I'll let you know with a bit of notice beforehand. The difference in distribution of these ions was a big part of why we saw this polarizing of the cell. Remember, a polarized cell is simply something that exhibits two different charges. Something that is more or less positive on one side than the other. And so the membrane is a barrier. Every cell has a membrane, and it's a barrier that can't easily be crossed unless we have something specific that allows a molecule to cross. We created that difference in charge by first limiting the ability of sodium and potassium to move. We separated them. Those ions can't move across the membrane. Sodium was outside, potassium is inside. And if you caught in the video, there are leak channels. Leak channels in the membrane. They are holes or pores that are specifically shaped for only sodium or only potassium. You think about those both as ions. They're probably little specks, but they do have individual shapes. And so one leak channel is shaped so that it allows sodium to move through. Potassium can't move through. But there's another set of leak channels that is shaped so that potassium can move through it, but no other ions can. So we have specific ion channels or leak channels in the membrane that allow movement of these ions through. And importantly, more potassium could leave than sodium could get in. More potassium Did I say that backwards? I think I did say that backwards. More sodium can leave than potassium can get in, which creates that differential. So if you have more positively charged things moving out, more of that positive charge moves out. Less of that positive charge is inside, and so there's a difference in charge between the inside and the outside of the cell. Not only that, but we actively move those ions. So we have leak channels that are holes in the membrane that allow things to move through. And whether they move, do you remember, this is not on the slide, but do you remember why an ion would move? There were two reasons. What are the two things that prompted an ion to move through that leak channel or not? Yeah, in the back. Pardon me? Diffusion, yes. Why would it diffuse though? Diffusion is the movement, but it could easily stay inside as well. You're right, it wants to diffuse. Why? Fantastic, that's one of them. Concentration gradient. An ion will move from a high concentration to a low concentration, absolutely. And so potassium wants to move out, sodium wants to move in. That's not the only thing though. If that were the only thing, it would be really hard to keep them separate. They'd always be rushing across the membrane along their concentration gradients. What was the other thing that affected whether they moved or not? Again in the back.
There's a large charge difference.
Absolutely, yes. The charge gradient or the electrochemical gradient. There's a negative charge inside which attracts positive things. So think about potassium. There's lots of it inside the cell. It wants to move out along its concentration gradient, but it's being held back because it's drawn to that negative charge. Those two things, however they change or however they're balanced, will make the ions move or not. Now that's getting complicated. We're not going to look at how these ions move in relation to those elements just yet, but those are the two things that would prompt an ion to move. So those two things dictate simple diffusion. but we actively move ions. We use energy to pump these ions. The sodium-potassium ATPase is something that if you haven't heard about it yet, you'll hear about it a lot in your academic career. It pumps 3 sodiums out of the cell for 2 potassiums in. It's probably responsible for using 20 or 30% of the energy that you're expending sitting here at rest. That's a lot. All of the cells in your body do this. All the energy you're consuming, a third of it almost, a quarter or a third, goes towards this process. And again, if you're pumping more positive stuff out than bringing back in, you're creating an inequality in charge. There's more positive stuff outside. Lastly, the thing that contributed to the difference in charge was that we had these larger negatively charged molecules that were stuck inside. Phosphate, PO4 negative, is bigger than just one sodium atom or one potassium atom. It's bigger. It can't fit through those channels. There are no leak channels for phosphate. Proteins within the cell, even bigger still. Amino acids linked together, folded on top of each other, a big glob of amino acids that does something in the cell. Those are negatively charged too. Those are stuck inside the cell. So we have a negative, or maybe a better way to put this is a less positive charge inside, because we have less potassium inside than outside, and more negatively charged things, phosphate, proteins. These are the elements that create the resting membrane potential. So I'm going to ask you another quick turning point question that builds on this a little bit. If you are inside a neuron and you're looking at the things in the environment, you see a lot of potassium. If some of that potassium started moving out, what would you experience? What would be the result of that potassium moving out of the cell as far as membrane potential is concerned? Would it have no effect? Would it make it more positive? Would it make it more negative? Would it depolarize the neuron? There are only so many ways that I can ask how it would affect charge. But if potassium moves out, what happens to membrane potential? Potassium that has a positive charge moves out, what would happen to membrane potential? Okay, good. C is correct. The majority of you got it. A positively charged thing leaving the cell takes away some of the positive charge, makes the rest of the cell more negative. It would make membrane potential more negative. It would hyperpolarize the cell. There's more of a negative charge inside. There's more of a positive charge outside. It would hyperpolarize or make it more negative. The odd option out here is depolarize the cell. That is a different way of saying item B, make the membrane potential more positive. B and C are saying the same thing. Okay. How is it possible the inside of a cell is negative? This is a fundamental question in trying to wrap your head around this idea. It seems weird that the inside of the cell has a negative charge, because after all, there's positively charged things inside, right? Potassium moves in. There's potassium inside. It has a positive charge, and there's lots of it there. How is it that the inside of the cell is negative? I think an important way to think about this is that maybe it's negative. Maybe the other negative things that are stuck inside overrule the positive potassium ions, but I don't think that's the case. I think a better way to think about this is that the inside is simply more negative than the outside. Or the inside is less positive than the outside. There's a difference in charge. It doesn't have to be negative itself. It's just less positive relative to the outside. Some of the things that contribute to that are the phosphate and the other proteins that are stuck inside. But the net charge, if you added up every single plus and minus sign of all of the things inside, the sum would be smaller than outside. So it's just simply less positive than outside. A way that we can illustrate that is if you can imagine doing a measurement. And we can do this with some microelectrodes. On the next slide, you'll see a schematic of what I mean. So this is a voltmeter. It's a voltmeter you might have if you were, maybe you're working on your car and you wanted to see if the battery was still good. You look to see if there's a 12-volt drop between the positive and negative terminals. This is something very similar. You're looking to see if there's a difference in voltage between one area and another. And those two areas that you're comparing are the starting point, which is the reference electrode. This happens to be outside. And you're looking to see how the end point, which is the sampling or the recording electrode, how it compares. And the two areas that we're investigating are divided by a plasma membrane. So we're looking inside versus outside the cell. In this example, the inside doesn't have to be negative. It's just less positive than the outside. And in fact, if you look in this cell, or in basically any excitable cell in the body, the inside is about 70 millivolts less than the outside. We've measured that. And it's slightly different in some cardiac cells, it's slightly different in some muscle cells, but on average, the difference is about 70 millivolts. The charge inside is 70 millivolts less than outside. Maybe it's not negative overall, but it's simply less than the outside. So, now that I've hammered that point to death, all we need to worry about is that there is a difference. There is the potential for ions, both positive and negative, to move. The membrane is a barrier that separates these two environments. It tries to stop those things from moving, but if we can put some checkpoints or control points in the membrane that we can turn on or off in specific situations, we can harness that movement. We can take that potential or the desire to move and we can link it to something. We can make it work for us. And making it work for us describes modifying membrane potential. We do this specifically. Resting potential in excitable cells in the body is on average negative 70 millivolts, or minus 70. 70 millivolts less inside than outside. And that might be something that I'd ask you on a midterm number 2. What's average resting membrane potential? Minus 70 millivolts. It doesn't stay minus 70. In fact, it changes wildly. How do we modify membrane potential? Well, remember, when we talk about membrane potential, the membrane that we're referring to is that of a neuron. And there's many different parts of a neuron. We reviewed them last class. The input zone, the trigger zone, the axon that sends a signal, if there is one to be sent. Membrane potential should be consistent at rest throughout that entire neuron. It should be minus 70 across the whole membrane. But we can make it different. And we do that by influencing or creating an input at the dendrites or at the input zone. Whatever the signal is, if we want to turn on or turn off a neuron, if we want to send a signal, we add a stimulus to the input zone. And that stimulus pushes membrane potential ever so slightly. It nudges it up or down. And that nudging is called a graded potential. So when you're measuring membrane potential and it's minus 70 at rest, if it moves to minus 60, you might be observing the action of a graded potential. It's slightly more positive. It might also make it slightly more negative. It depends on what signal is being sent to this neuron. A graded potential will push it up or push it down. It generally diffuses into the body of the neuron, but it doesn't go very far. It makes small localized changes in membrane potential. But if we add a lot of these graded potentials together, we have the option or the opportunity to create a large deviation in membrane potential, to create a big signal, to turn on or turn off a neuron. If all of the graded potentials added together affect resting potential enough, it might trigger the axon hillock to send a signal. It might light up that nerve. If that happens, then the neuron is on. It sends a signal. The signal has the strength. Well, when the signal is activated, the signal propagates all the way down the axon and it doesn't lose strength. The signal goes to completion through the rest of the neuron. If the axon hillock is stimulated, that signal is called an action potential. We've mentioned action potentials a few times. I've even probably said it in passing. We send an action potential to muscle. Well, that's a signal to contract muscle. I got that signal from somewhere. The motor neuron that attaches to muscle was triggered, and it sent the signal all the way down the axon. That is the action potential being set. So we're going to learn about graded and action potentials today. Graded potentials, small little deviations in resting potential. Action potentials are large signals. So I'm going to ask you another question quickly. In relation to a graded potential, your understanding of a graded potential thus far, which of the following would not be true? Can a graded potential make membrane potential more negative? Can it make it more positive? Is it received at the dendrites? And does it trigger an action potential? Which one of these is not automatically true? Which one of these is not automatically true? If a graded potential is registered inside a neuron, which one of these won't automatically happen? Okay. So maybe this is overloading you with information or not explaining it well enough. But the answer is D. It doesn't automatically trigger an action potential. It can push membrane potential up. It can push it down. If the sum total of all of those nudges is enough, it will trigger an action potential, but it won't always do that. It is definitely received by the dendrites and spreads locally through the body of the cell. So keep that in mind. We're going to look, rather than me describing it to you, we're going to look at how the signal propagates in another animation.
Membrane potentials, graded and action potentials. describe the generation of changes in membrane potential, we will use a motor neuron, which stimulates skeletal muscle to contract. The neuron can be divided into three zones. The input zone, consisting of the cell body and extensions known as dendrites, has chemically gated channels in its membrane to receive stimuli from other neurons as graded potentials. These graded potentials move toward the trigger zone at the axon hillock. The trigger zone is where voltage-gated channels are first encountered in the neuron. If the graded potentials reach threshold in the trigger zone, then an action potential is generated. The action potential is transmitted by a voltage-gated channel down the axon to the output zone, where it causes skeletal muscle to contract. In the input zone, graded potentials are generated when stimuli such as neurotransmitters attach to receptors of chemically gated ion channels, causing them to open briefly. Then, shift in the distribution of ions on either side of the membrane results in a slight change in electrical potential. Graded potentials travel a short distance through an input zone toward the trigger zone before fading away. It is the net voltage produced by graded potentials at the trigger zone that determines if the threshold of voltage-gated channels is reached to trigger an action potential. Some graded potentials occur when chemically gated sodium channels open to allow sodium ions to diffuse into the input zone. This electrically moves the trigger zone closer to or above the threshold. They are considered to be depolarizing graded potentials. If chemically gated potassium channels in the input zone are stimulated to open, it allows potassium ions to diffuse out of the cell and causes the inside of the membrane to become more negative, moving the voltage in the trigger zone away from the threshold. These inhibiting graded potentials are known as hyperpolarizing graded potentials. Since depolarizing graded potentials cannot individually reach threshold, an insufficient quantity does not cause a response at the trigger zone. If, on the other hand, there are enough of them to reach threshold, an action potential is generated. If there are more hyperpolarizing graded potentials than depolarizing, the net membrane potential in the trigger zone becomes even more negative than the resting potential. This would temporarily inhibit the generation of an action potential or require additional positive stimulation to reach threshold. When the net graded potentials arriving at the axon hillock reach threshold, the
That's all we care about for now.
membrane potentials, graded.
So in the video, you saw the puff of green cloud. That's a wave of positive charge. The puff of red cloud was a wave of negative charge. And so a green cloud makes the membrane potential more positive. A red cloud makes it more negative. And if you have enough of that green cloud wave propagating through, you might be able to trigger a signal in that neuron. So the graded potentials are the way that you turn the neuron on. They're small, temporary deviations in resting membrane potential. You receive them at the dendrites, they spread through the cell body. Some of them, like you saw in the video, open potassium channels, potassium rushes out, the inside becomes more negative, and it inactivates the neuron. It prevents it from turning on. This is what we call a hyperpolarizing graded potential. Whatever the signal was, it opened up a potassium channel that allowed potassium to escape. Positive things leaving makes the inside more negative or hyperpolarized. Some of the signals that arrive won't target potassium channels. Instead, they'll target sodium channels. And if you open a sodium channel, sodium rushes in. Remember, there's lots outside and very little inside. It will rush in, bringing positive charge into the cell with it, and the membrane potential will go up a little bit. This is a depolarizing graded potential. And if one of these arrives at a time and that's it, this is all you'll see. You'll see a brief hyperpolarizing potential, or you'll see a brief depolarizing potential, and it will be restored to normal. One at a time is easily dealt with. No signal occurs. No larger signal than this, at least. If many arrive, we have the ability to make something happen. First, whether a graded potential happens or not, we can change the strength of that graded potential. You might have heard in the video that a graded potential arises from the opening of special channels. These are mechanical channels, or what are called ligand-gated channels. Ligand simply refers to a molecule that binds to it. A ligand is a neurotransmitter, like epinephrine, or acetylcholine you might have heard of. A neurotransmitter is a ligand, and it will bind to one of these channels and open it up. So, if we want to make the graded potential stronger, there are two things we can do. We can open up more channels, or we can open the channels longer. Both of those has the effect of allowing more movement of ions. More sodium rushes in. More positive charge comes in. It's more depolarized. So we can increase the likelihood the neuron will turn on by increasing the strength of the graded potential. How many channels open and how long those channels are open. But even still, the strongest graded potential is unlikely to trigger an action potential. How do we ever send signals with neurons then? If the largest graded potential is not enough to trigger that neuron, how do we ever trigger that neuron? We overload the neuron. Each of those conditions was one graded potential at a time. If you send one signal and register one graded potential, it can be dealt with and restored back to normal. But what if you sent 2, or 3, or 10? If you send a second graded potential before the first one has been resolved, it's additive. So imagine you're recording the difference again with your mini electrode. The resting potential is minus 70 millivolts. You register a depolarizing graded potential, a positive wave. And then you see a second one occur. If a second graded potential occurs before the first one has been resolved, it's additive. That is, it will still increase membrane potential on top of wherever the new starting point is. Now we can start to see a way that we might ratchet up the activity of a neuron. If we sent a lot of graded potentials, we could raise that potential, raise the charge to a certain threshold. And that idea of threshold is really important. That idea of threshold is important because we don't want our neurons firing randomly when we don't want them to. You don't want to be sitting there and then jump out of your seat for no reason. It would be embarrassing, right? Threshold is like a fail-safe. Only when you reach threshold will the neuron activate and will an action potential be sent. So let me introduce this concept of an action potential and then we'll call it for today. only when you have a large enough signal from the combined effects of the graded potentials delivered to a neuron will an action potential be generated. So an action potential only results when the graded potentials cause the charge to reach threshold, whatever threshold happens to be. Ladies, have a question? Is this not clear? Remember what threshold was? Yeah? What's threshold? What's the idea of threshold? It's the point at which you would activate the neuron. Nice job. Good collaborative effort. Absolutely. So if the membrane potential reaches threshold, you would activate the neuron at that point. When you activate the neuron, when you generate an action potential, it happens. There's no nudging this way or that way. There's no little deviation in charge. It is an abrupt, large change in the charge of the neuron. It occurs rapidly. It is a disruptive change in membrane potential. It causes a large spike in the charge of that cell, and we'll see that coming up. Membrane potential, where it's normally negative 70, actually becomes positive. It jumps to positive 20 or positive 30. Think about that. Using your microelectrode, your recording electrode, normally the inside is more negative than the outside. During an action potential, it flips. The inside becomes more positive than the outside. We don't want that. Certainly not long term. And we're doing our best to pump ions back to maintain normal resting potential. But during the action potential, it shoots up drastically and quickly and becomes positive. Eventually it's restored, it's repolarized, but there's no way around it. If a neuron is triggered, if an action potential is sent, all of the neuron depolarizes. That's what's meant by this all or nothing item on the list. A graded potential might have an effect or might not. The more potentials you have, the more of an effect you have. An action potential is an action potential is an action potential. It has one strength. It is turned on or it's not in response to whether or not you reach threshold. So we'll continue that animated video when we get back to class on Tuesday. You'll see what happens in the membrane when an action potential is triggered. Otherwise,